So I'd like to welcome you here today. Thank you for your patience with the issues with logging in. I appreciate seeing 74 people here to learn about CompuCell. Today is going to be a very gentle introduction to CompuCell. So if you've used it already, this should be material that's familiar to you. If you have not used CompuCell before, then it will be a bit new, probably. And if you're an expert, please think of yourself as a TA today to help those who are getting started. Remember the days when you were getting started. As always, I need to remind people that this meeting is live streamed and recorded. As always, I'd like to thank our funding sources, the NIH and the NSF, which have been very generous in creating the CompuCell project. So today we're going to introduce the course and its logistics, people, schedule. We'll talk a little bit about the hackathon. Somebody asked yesterday, do you have to do the hackathon? No, you don't, but it's a great way to learn and consolidate things, especially for beginners. And one of the options in the hackathon is to duplicate existing simulations. So you don't have to do something brand new to get benefit from the hackathon. And if you have a lot of biological experience, for example, haven't done modeling before, that can be useful too. I'll give a little bit of introduction to virtual tissues. We'll talk about that more in the second session. And then we'll jump in and actually run CompuCell. We'll run the angiogenesis simulation. Some of you already did that yesterday. We'll play with Player a little bit and learn how to use Player. And we'll talk a little bit about how to download files, open them, and start using Twedit, the model editor for CompuCell. And then I'll make a few comments about debugging. CompuCell is a complicated package. It can throw errors. Some of those errors are relatively straightforward to fix. Other ones are less straightforward to fix. And I'll talk about a couple of the problems that you might run into more commonly that are a little bit less obvious to debug. So logistics of the class. We're going to use the Zoom. Please keep your mics muted when you're not speaking. Put your questions in the chat. There'll always be somebody monitoring questions in the chat. Feel free to use the comment buttons if you think we're going too fast, if you need help, and so on. We'll do our best to monitor things. All of the demos and exercises are available through the Google Drive. So please look for those, and the videos are on the YouTube channel. People, we'd like to thank Hayden Fennell, you all know here, who stepped up to be the lead organizer this year. That's a big job. We really appreciate him doing that. Everyone here pretty much is a volunteer for this project. We have instructors. Priyam Adyapok from Duke University is going to give some lectures. Giulio Belmonte will be speaking today from North Carolina State. Pedro Castel, who's a grad student here, is an expert in CompuCell, has been helping, and he'll be lecturing. TJ Segal from the University of Florida will be giving some lectures. Jim Sluka, senior scientist here, will give some lectures. Gilberto Tomas from Brazil will give some lectures. Lorenzo Vischini from London is helping out and will give some lectures as well. And then there are a number of additional people who volunteered their time to help out from time to time as either guest lecturers or TAs. Again, if you have any questions or problems, you can reach out in the chat, in the Slack, or Hayden Fennell's email, hfennell with only one L at the end at iu.edu. We have some additional guest speakers on the later parts of the class. Megan Haas, Tom Knudsen from the EPA, Calvin Lam and Cheng Zhang are going to be giving extra lectures as well, showing some of the things you can do with CompuCell. The daily schedule, we're going to have a half an hour before we get started for things like dealing with Zoom logistics or questions or comments from the day before. We'll have about a 1.5 hour module, then a break. That first break may get eaten into a little bit if we have delayed starts because of logistic problems. Second module, a midday short extra session, which we'll talk about on days each day. The lunch break, 2 modules, and then time for questions. We realize that that's a long day. Last year, we taught this in 2 weeks. It meant that we only had to have 3 modules a day, but it also meant really a long time commitment for everyone. And so we're trying to get it into a slightly more compact form this time. For those of you who are in bad time zones like China or India or Europe, we appreciate your patience with us staying up late when we need it. Today, we're going to be doing CompuCell basics. We're going to start out just playing with CompuCell a little bit. I'll give you some examples of virtual tissue applications next, and we'll look at the code just to get a sense of it. And then we'll have some demos from instructors of different things you could do with CompuCell, a little more detail. Julia will then get into the basics of using CompuCell, and we'll have some exercises to explore different aspects of CompuCell in the afternoon. Tomorrow, we'll start actually getting into principles of modeling and detailed modeling instructions. Wednesday, chemical field, secretion, uptake. Thursday, chemotaxis. Friday, I'll teach you how to combine network models with CompuCell if you want to do things like calcium waves. We'll have some hackathon preparation in the middle of the day. Saturday, we'll talk about compartmental cells and more elaborate biomechanical models, and that will continue on Sunday. Some of those things may change depending on availability of individuals and timescale. I want to talk a little bit about the hackathon. As I said, the hackathon is completely optional, but people usually have a really good time with it. It's a great way to learn. A couple of years ago, some people actually started a company based on what they did in the hackathon. So anything is possible, but you can be a beginner and still get a lot of benefit working in a hackathon setting. And we have plenty of ideas and suggestions for things that people can do. We will start at the end of this week talking about hackathon teaming. We'll have meetings during the week next week, and then we have the hackathon, not this weekend, but the following weekend. This is a bit of an experiment trying to understand, give people a little more time to consolidate their CompuCell knowledge before they jump into a hackathon project. So there are going to be some documents, and Hayden will explain the hackathon ideas a little bit more. I do hope people will decide to participate. Again, it's totally optional, but I think it's a lot of fun. You'll be sleepless for a couple of days, that's true. But if you really think you might want to use CompuCell seriously, it's a great opportunity. We have a lot of online resources. CompuCell resources, there's the CompuCell website and the downloads. The online manuals are very good. We really encourage you to use the online manuals. I would ask everybody I understand if you only have one screen on your computer, it's a little more difficult, but I really encourage you to have the online manuals page up on your computer so that you can reference it all the time. There's a quick start guide, which is rather out of date. It's from CompuCell 3.6, but a lot of the things in it are things we're going to do today and haven't changed. The class has a Slack channel, a Google Drive. The CompuCell website may not be updated quite as fast as the Google Drive. If you're interested in Antimony and Tellurium for networks, there's a nice web page for that. Hayden will share the links in the chat for all of this. And there's also a nice quick reference guide to CompuCell. The Slack channel, I think people, if people need help getting into the Slack channel, please put it in the chat now. It's a great way to help each other. The other thing which I don't have on this slide that I mentioned before, and I'll mention it again here, is that ChatGPT and Claude are very, very good resources for Python programming. And ChatGPT at least knows CompuCell XML and CompuCell Python. So if you get stuck and you would like help, I encourage you to ask ChatGPT or Claude for suggestions for algorithms and ideas. Of course, those AI systems do make mistakes, so you can't automatically trust the things that they do. The good thing with AI-generated code is that if you try it out, it works or it doesn't work. And so you generally know pretty fast if it's making a mistake. One thing I found with ChatGPT generated CompuCell is that the algorithms it generates tend to be pretty slow. They tend to work, but they are a bit slow. But Hayden was introducing the idea of classes, for example, yesterday in Python. It's a somewhat sophisticated Pythonic concept. ChatGPT does know about Python class structure in CompuCell. And so if you need help with that, actually, it's a pretty good resource. In the old days, you'd go to Stack Overflow to look these things up. ChatGPT basically has read Stack Overflow and indexed it for you. So I do encourage you to use ChatGPT. I wish somebody could write a little extension to Wizard that would pull in ChatGPT suggestions. A lot of the IDEs now, like Visual Studio Code, have AI-assisted code completion. It's pretty helpful. The quick reference sheets, I encourage you to print them out. There is a general Python introduction that's about 5 pages. We really only need the first 2 pages of it. The key things you'll need from Python are the idea of loops, links, lists, and dictionaries. A little bit on functions. If you know those 4 things, you pretty much know the Python we need. There's that CompuCell3D quick reference guide, which is worth printing out, and the Tellurium quick reference guide for network modeling. If you're interested in learning a little bit more, there are a couple of book chapters and papers going into the internals of CompuCell. Magnetization to morphogenesis really teaches the physics of CompuCell, if you want to learn that. Introduction, single cell-based model book has some pretty good things about this kind of modeling in general. If you have never built mathematical models, it's not about agent-based modeling. It's more about ODE modeling. There is a paper that was written by some Canadian graduate students a few years ago called 10 Simple Rules for Attacking Your First Mathematical Models, a guide for graduate students by graduate students that is pretty good at introducing a lot of these ideas. So those are resources that are in that student materials page that you might want to look at. Almost nobody comes to this course knowing everything that they need. We've had everything from high school students to emeritus professors, everything from pure mathematicians to surgeons to computer scientists to physicists. One thing that's nice about having a big group is that people can fill in each other's knowledge gaps. We like the Rosalind online Python exercises. For network modeling, that online course is very good. There's also a great book, Systems Biology, Introduction to Pathway Modeling. For basic mathematical biology, Essential Mathematical Biology by Britton. It's a great short introduction to basic concepts. For developmental biology, which is very important for what we'll do, I love Gilbert, Developmental Biology. The old editions are about $2 on Amazon, and they have almost everything you need. Wikipedia is pretty good in biology. For physics, the Phil Nelson's Physical Models of Living Systems is a good, gentle introduction to the physics of biology. the Rob Phillips Physical Biology of the Cell is a beautiful textbook, which I taught out of for many years, but it's pretty advanced. If you're not really a physicist, it's hard to read. There is not a textbook on CompuCell type modeling yet. There needs to be. If somebody wants to help us write it, we would appreciate your help. We've suggested some papers. The online manuals are pretty good. If you find mistakes in the manuals or have suggestions for how they can be improved, gee, this is unclear. Gee, we need another exercise. Gee, we need another demo. Please screenshot it, make a detailed suggestion, and send it in, and we'll do our best to improve. Okay. There is a book on cellular POTS models. It's a little bit old, by Shanna and Preziosi, and that's worth looking at, although things have changed somewhat since that. If you have sources, review articles, online materials, anything you think that would be good to help people learn, please send them in and we'll add them to the stack of reference materials. Okay, why do we need to do multicellular modeling in the first place? You probably have some idea why you want to do it, or you wouldn't be here. For me, ultimately, I would like to be able to see us make medicine more of an engineering discipline, more of a scientific discipline. At the moment, essentially, you wait till you're sick. When you're sick, you go to a doctor, a hospital. You wait a long time for diagnosis. They do treatment. There's certainly some feedback. They certainly do some adjustment of treatment while you're in the hospital, for example. But it's pretty common the doctor will say, take these pills, come back in 3 weeks and tell me how you feel. We'd like to be able to close that loop and actually regulate health rather than just respond to disasters. To do that, you need to be able to predict what's going to happen, both with and without treatment, to do optimization of treatment. At the moment, most medical treatment is still not personalized. It's not what is now called precision medicine. It's best for the population, not for individuals. We often don't know if treatments are working. We can't, of course, evaluate alternative treatments in a single individual. You either treat or you don't treat. You can't say what would have happened if you'd done something differently. And you can't really adjust treatments dynamically, except in limited cases. And so computer prediction of health state is going to be critical. Now, whether the methodologies that we're going to use here are going to find their way into the first generation of medical digital twins, I can't tell you. Computationally, they're pretty heavy, and so they may not be in the first generation of medical digital twins. What we can do is a lot of basic biology and bioengineering. We can understand how organisms develop, how they maintain themselves, because maintenance is not trivial in living systems. We can understand what are called developmental diseases, how the things that build and maintain organisms fail. The classic example of that is cancer. We can look at infection and immune response. We can look at bioengineered tissues or how cells behave in a dish. Something that we use this kind of modeling for a lot is toxicology. How do chemicals perturb biological function at different scales? All of these problems involve an interplay between physical and biochemical mechanisms, in particular biochemical network control and biophysics. And modeling is really essential to understanding how systems work. We could call that multicellular systems biology. We start out with the basic concept of a cell, an extracellular environment, and we can build an incredible diversity of tissues with different structures and functions. On the other hand, if we look at what single cells do, well, they can grow, divide, differentiate, die, metabolize to build things, produce waste products and energy. Cells can mutate, change their behaviors. They can move. Cells can interact with each other, to stick together, to pull on each other, to eat each other, to send signals to each other. They can interact with their environment by secreting and absorbing chemicals. They can send signals in the environment. They can pull on the environment. They can build solid materials in the environment, and they can create and move fluids in the environment. There's some basic hard limits in physics. Diffusion is a critical biological process. Mechanics limits what can happen. And if you put those together, you get complex structures. One of the things that people have worked on a lot with this kind of modeling is cancer, in particular, why you get what are called the hallmarks of cancer. For an initial cell to become a cancerous cell, it has to make many changes to itself and its environment and its neighbors. Those changes are quantifiable and repeatable, even though the individual subchanges are not, and one can understand why that is using this kind of model. What are virtual tissues? That name may not be the most useful. It was invented by the EPA. We liked it. We could call these dynamic agent-based multicellular models, if you prefer. That's what we used to say. It's a longer name for the same thing. We're looking at emergent spatiotemporal phenomena. If there's no spatial structure, you don't need this kind of model. We're focusing on interactions between cells and cells, between cells and chemicals in their environment, and between cells and the mechanical properties of their environment, and so on. These are mechanistic models. They're additive. And this is tricky. Nothing exists in our world unless we say it exists. We don't get anything for free, or very little for free. That means we can't take for granted that things exist. In the real world, we get physics for free. In computing, we have to build that in. Our models are going to be based on models of object behaviors and interactions. And typically in the simulations we're going to be modeling hundreds to tens of thousands or hundreds of thousands of cells. Think about what you see in about a 20x microscope objective, and we'll get movies. And we have to think about what to do with those movies. How do we get meaning from them? Why do we need this? Well, if we think about a drug acts at a molecular level inside of individual cells. When we make medical measurements, we typically are looking at whole body results, the pulse, blood oxygen level, biochemistry in the blood. So we're measuring at the whole body level. But disease or normal function really play out at the level of tissues and organs. And so there's a disjunct between the level at which we can make measurements, which is very coarse, the level at which we can control, which is very fine, and the level we care about, which is in the middle. And if you're an engineer, you learn from the beginning that you should measure and control at the same scale, and that scale should be a scale you care about. And so it's really a problem medically and from an engineering point of view that the scale at which we can control is separated from the scale at which we can measure, and we're both separated from the scale that we care about. And virtual tissues as multiscale models can help us connect those scales together. I always use the same example, which is if I gave you a list of the parts of your car, this screw, this spark plug, this battery, and then I asked you to tell me, what is the car like to drive? You couldn't tell me. The emergent behaviors of the car depend on the parts, but they're not a result directly of the parts. That emergent behavior isn't something you can predict from the quality of the screws. And so we're in that situation in biology, but it's even more complicated. One of the things that we face in biology, especially in development, but also in things like wound healing and cancer, is that there's a lot of movement. Cells reorganize, tissues reorganize. Here we have branching morphogenesis. Here we have a movie of all of the cells in a developing zebrafish embryo. This is a detailed model of blood vessels growing in between what are going to form the segments of a zebrafish embryo. And this is a chicken, which is forming its periodic segmental structure in the backbone. A lot of change, a lot of morphological change. And that morphological change really is the heart of the kind of modeling that we're going to do. We're going to start out at the middle with modeling individual cells, what those cells do, how they interact with each other. We're going to talk later in the week about biochemical control, signaling, gene regulatory networks. We're not going to do much in the way of metabolic networks, but the methods are going to be the same. We're going to put those cells, in this case, we actually, on the right-hand side, we actually have biochemical oscillators inside of every cell. Those biochemical oscillators communicate locally between the cells. There are long-range chemical gradients in the middle. And based on those, the cells differentiate, change their mechanics, move around, and change their properties. Often, we also add another layer, if we're doing medicine, which is we'll build what's called a pharmacokinetic pharmacodynamic model, PBPK model, of the absorption, distribution, and elimination of drugs in the body. That's typically not a spatial model. They're pretty standardized. But if you want to know when you take a pill, what's the level of chemical of a particular drug in a particular organ at a particular time, you use that kind of model. And you can then open up a box like the liver, put in a detailed multicellular model there. Inside those liver cells, you could put a model of the metabolism of the liver cells. And I'll show you some examples. This kind of modeling has been used to understand basic biology. It's been used to predict both positive effects, drug effects, and adverse effects, toxicities. It's been used to design new therapies, to match therapies to individuals. And it's particularly useful when you have movement and reorganization of tissues, where you actually have to think about the spatial organization of tissues. Virtual tissues are pretty powerful. You can change the level of resolution of the imaging you do. You can do unlimited tests at different doses, for example, of a drug, and ask when feedback either protects a tissue or amplifies an effect. You can test hypotheses. It can reduce the need for animal experiments, which is a big issue these days in terms of ethics. It can allow you to test things that would be very hard or impossible to do experimentally or impractical. You can certainly do many more combinations computationally than you could do in an experiment. It's also a good way of communicating. The movies you get are very immediately meaningful to people, so it's a good way of communicating. And it's a good way to test your knowledge. These kinds of models are not trivial to build, and they're remarkably good at revealing what you don't know about biology. You can build counterfactuals. You can create situations which are not possible in nature, and that's particularly useful if you're going to train AI systems. AI based on real-world data suffers from two problems. There's no gold standard in medicine. You don't really know what you're putting into your AI systems, and you cannot put in things that are very rare or don't happen. With simulations, you can generate those, and you know what you got. And so for training AI systems, mechanistic models are absolutely essential. And so there are a whole variety of reasons that this is useful. It's not easy to do. Biology is complicated. There are a lot of components. There are a lot of parameters. We don't know their values very often. Components, unlike two screws of the same kind, which are really pretty much the same, two cells of the same type are not. And so we have to worry about that. Cells change their behaviors. Screws may break, but beyond that, they don't really change their behaviors. We're always in a phenomenological world. We never have complete detail, and we don't always know exactly how to compare experiments with simulations. In particular, with stochastic models, every time we run the simulation, we get a different result. And so we need to think about ways of comparing those. That's something we're not going to address so much here, although TJ Seago is working on that in his own research a lot. People might ask him about that. There's a lot of missing scientific knowledge still. And there's a huge range of length and timescales, which we can't include completely. If you want to do it anyway, you can ask how. There are a lot of different ways you can represent the things in your models. And there are also a lot of frameworks now that'll help you build, run, and share them. This is a list of some of the frameworks. We're going to be talking about CompuCell3D. Morpheus and Artisto use the same basic methodology as CompuCell. Physicell, for example, and TissueForge are what are called center models. So it's chased, really. And there are other modeling methodologies. All of these have their advantages and disadvantages. CompuCell is probably the oldest of them, or one of the oldest of them, and one of the more widely used ones. And we hope it's one of the easier ones to use, that you'll find that today. Just in terms of representing cells, we have what are called center models, where cells are basically points. This is like a molecular dynamics simulation. There are a lot of different center model packages. Center models are great because you can have lots and lots of cells. They run very fast, but they don't have a lot of spatial detail. If you want explicit cell shape, there are a lot of different ways of doing that. You can really use finite element solids type models, sort of abacus-like models. Those tend to be computationally very expensive, but you get a lot of spatial detail. If you're thinking about sheets of cells, what are called vertex models are very effective. Multicellular element models or subcellular element models basically cluster the point-like particles together to make essentially represent cells as if they were molecules. They have some of the advantages of center models. We're going to be dealing with this kind of lattice-based modeling today and during the rest of the week. As I've said, there are many frameworks now available. We're going to be using CompuCell. It's what we've developed for the past 20 years. It's a very flexible way of building simulations. It has some big advantages in the sense that it's got pretty good support for model building. It has some limitations, and we can talk about those later in the week, depending on what people's particular interests are. It allows you to do network models very easily and integrate them with the cell models. It uses the CPMGGH lattice method to describe cells in 2 or 3D. Julia will talk about that in more detail a little bit later today. Subcellular network models are pretty easy. You can build chemical fields and their environments. It has some limitations. It doesn't do advection very well. It doesn't have explicit biomechanical fluids and solids models built into it. You have to couple it to other packages. But it is also a way of building and sharing models pretty easily. And you can, in fact, execute models online. As I mentioned, there's this web version that on NanoHub, it's a little out of date, but it still works. And you can certainly run these models on Amazon Web Services, on Colab, and elsewhere once you have them. Basic concepts in CompuCell. Chemical fields, that is concentrations of things in space. Concentration, diffusion, decay. And they can interact with cells. They can react with each other. They can be secreted, absorbed. You have cells and what are called generalized cells because you can represent just volumes of material. For example, the nucleus of a cell, the cytoplasm of a cell, leading edge of a cell. They have basic properties like volume, polarity, surface area, and behaviors, movement, growth, division, death. They can interact by sticking to each other, responding to external chemical environment by moving, differentiating, secreting, and absorbing chemicals. Both fields and cells are fundamentally rather fluid-like. To represent solid processes, solid materials, and also to represent structures like filopodia in cells, we have a concept of links. These are essentially little springs that you can attach between cells or the components of cells, and they allow you to build materials with very specific biomechanical properties. And they're really designed for force generation in complex situations. And at the end of the week, you'll learn a lot about links. And then, as I mentioned again and again, we have a concept of networks. These could be gene regulatory networks, PBPK networks, signaling networks, or metabolic networks. And those can be implemented either as ordinary differential equations or as Boolean networks. And here's some examples of the kinds of things you can get with that. You can have a cell cycle oscillator that controls the growth and division of cells running inside of individual cells. You can have what's called delta notch signaling. Lorenzo's a big expert. One of our TAs is a big expert on that. You can couple those together, and we'll definitely do the delta notch model in class. TJ may do that MaBoSS Boolean model in class. We're thinking about teaching a course on specifically modeling the immune system with Boolean network modeling in the fall if people are interested in it. Okay, so that's enough of an introduction now. We're a little bit behind because we started a little bit late. I'd like everybody to just run a simulation, make sure the CompuCell is working. We're going to be running a little angiogenesis simulation. Some of you ran this yesterday. Please follow along with these exercises. We'll build this simulation from scratch later in the week. At the moment, we're just making sure that it runs and getting familiar with CompuCell. Any questions before we get started? So please, everyone, get CompuCell running on your computer. Launch CompuCell. So to begin with, we're going to run a CompuCell simulation. CompuCell comes packaged with a bunch of demos. We're going to open one of them and run it. CompuCell has two basic tools. If you're using it in normal mode, if you're very sophisticated and running it in Jupyter notebooks or in Visual Studio Code, this is not going to apply to you right at the moment. It's got a model editor called Twedit and Player, which is a GUI for model execution. And these are software to help you run and write models. These are not models themselves any more than Microsoft Word is a letter or a model. And let's follow along together. We're going to run an angiogenesis simulation. This was a work done by Roland Merks when he was here in Indiana many years ago. He's now a full professor in Leiden University in the Netherlands, based on work that he was doing trying to understand how blood vessels develop. And the angiogenesis simulation is very beautiful. It's a very simple simulation, but it's very powerful, and it represents some very fundamental biology. If we were interested, there are a series of papers from about 15 years ago now, which explain it in some detail, 15 to 20 years ago now. We are trying to understand how blood vessels organize. And one thing that's interesting about blood vessels is that the same kinds of cells, endothelial cells, can form new blood vessels in two very different ways. You can have an existing blood vessel, which can sprout, send out shoots, like a tree sending out a branch, or you can have blood vessel cells that are initially completely separated from each other, like raisins in bread, that come together to organize to form capillaries. And we were interested in knowing whether the same mechanisms could explain both of these things. There were experimental observations, which were analyzed by Gamba and Serini about 20 years ago, that found that in vitro, when you put the cells in a dish, cells migrate to higher concentrations of cells. They couldn't visualize the chemicals directly, but they suggested that the cells were secreting a chemical which attracted other cells. That's called autocrine chemo or paracrine chemotaxis. They also noticed that if you put in too much of a particular growth factor, vascular endothelial growth factor A, cells stop moving. So that suggested that might be the signal. And then experiments by our collaborators at University of Kansas Medical Center showed that there was one more thing you needed. When cells touched each other, normally they don't respond on the surface where they touch each other to the chemical. And if you prevented them from what is called contact inhibition of chemotaxis, they didn't form blood vessels, they formed blobs. And so Roland built a simple simulation of that where you have cells. Here you see the color is the concentration of the chemical. The cells are attracted by the chemical. If they respond on all of their surfaces, on the left, they form blobs. On the right, they formed a vascular network, very much like what happens in experiment. So you have cells secreting a chemical, which diffuses. They move up a chemical gradient, and they stick to each other. That's all they do. So now we're going to use CompuCell to run that simulation. On Windows, search for CompuCell3D in your search tool, launch Player. On Mac, you'll have to find the CompuCell folder and execute the CompuCell batch to get it running. First time you launch CompuCell, it may take a while. Don't close the terminal window. Don't give up. If you're doing this the first time, you may have to provide execution permission for CompuCell to launch. So we're now going to explore Player a little bit. Don't worry, we will do this again and again. You're going to hear these things many times. I always say the same thing, which is if something is worth doing once, it's worth hearing twice. So we're going to go over it a couple of times. For people who are familiar, I appreciate your patience, but remember, we were all beginners at one point. And people who are beginners, don't be shy about reaching out for help in the chat. Okay. When you use Player, you'll see a window that looks like this. If you're on a Mac, it'll look a little bit different, but this is the basic window. It works like a normal window. You can resize it with the corner. You can change the size of the bounding boxes within the Player layout to make it a little bit more convenient. On modern high-resolution 4K monitors, the size of the box buttons and things may be a little bit small to see. So the first thing we're going to do is open a simulation in Player. And so you should have Player running. And you want to go to the File pull-down menu and select Open Simulation File. And you should pull up an open file window. And you're going to navigate to Demos, Book Chapter Demos, Angiogenesis. At the moment, I'm not asking you to open the file. I haven't gotten that far. You just have to find it. Once you have found that, if somebody says they went ahead and clicked on the file ending in CC3D, yes, that would be good. So let me actually do this. Okay. Here is CompuCell Player. And I realize that it's not going to be so easy to see with the screen resolution that I have here. If you hit this button, this up arrow button here, can people see that? That will do Open Project, or I could go to CC3D Project, Open Project. Does the dialog box open for you? I'm going to go to MaBoSS, my C drive, CompuCell3D Demos, Book Chapter Demos, Angiogenesis. And now I'm going to select angiogenesis.cc3d and hit Open. This is actually in Twedit. I wanted to go I wanted to do that in Player. So let me switch to Player. So this is Player. I'm going to go to File, Open Simulation File, OS, CompuCell3D Demos, Book Chapter Demos, Angiogenesis, Angiogenesis.cc3D. Oh. Once you have this working, you can hit this circle button here, that's step. If you hover over it, it'll say step, and you'll see the simulation run. If you hit this button here that's labeled play, the simulation will continue. The two dots are pause. Don't hit the stop button unless you need to. Again, pause, stop, run button here with the little arrow after an old CD player or tape recorder. The single circle, which would be record on an old tape recorder, is step. The pause button. The stop will reset the simulation to the beginning. So in general, you shouldn't reset the simulation unless you know that you don't want to continue it. Hit pause rather than stop. Okay, now that you've got it running, you can actually start playing with visualization. The default, if you haven't played with CompuCell before, is that you'll see something that looks like this on the right, which is a 2D image. And you may want to resize that image. You can use the full screen box, which will work, or you can pull the bottom corner of the box to resize it. There's also a very convenient tool in Windows only, which is Tile, which will help you lay out the screen, which we'll come back to. You're seeing a 2D section. It'll say XY here, a 2D section of a 3D simulation. What I want you to do is pull down, go to XY, pull that down, and select 3D. And after you've changed the display settings, you will have to hit run again. It will pause when you hit the when you pause the display settings. So let me come back to that. I'm going to do it with you. Okay, here is what you would get when you start the simulation. XY 25, step it. I'm going to hit pull this down. And one problem that can happen, it may put the selection in a funny place when I move the screen. So the problem for me was that I had moved the window. So if I do it here, it's there, but it's hidden. It's there, but it is having an issue in my screen because of the zoom share with pulling it down. Pull down XY, select 3D. To get that window to appear, to pull down to appear, you have to at least done a single step. So if you just launch it, you have to hit one step or hit run before it will work. And now hit play again. As you have the simulation running, you can use your mouse over the simulation to zoom in or out, to rotate the simulation. And if you get a setting that you don't like, if you hover over the image, you may have to look for where that pull-down menu appears, depending on how your screen is configured. It's there, but it may be hidden. So let's come back that again. Hit step. You'll see this. Pull down the XY. You may have to look to see where it displays the little choice menu and select 3D. Somebody says, I see the coordinates, but it was not the simulation in 3D. If you have the simulation, it's possible to adjust the screen settings so that you lose the simulation image, Typically, if you hover over the simulation image, hit the letter R, it should reset the visualization. It's also possible if you pull down the window menu and you're on a Windows machine, hit Tile. That's a very useful function. That'll lay out your screen and put things in a reasonable condition for you. Try that. And if things get really messed up, try Restore All Graphic Windows. That also should help reset things. Player offers a lot of visualization commands. Those are mostly available in the pull-down menu labeled Visualization. In particular, you can select whether to represent the cells. If you turn the cells off, you don't see anything pretty much. Whether you're going to represent their borders, whether you have any 3D output. There's some other things that we're not going to use so much. When you change the settings, you have to remember that the simulation will pause. And so you'll have to hit step or start after you've made a change. The other thing that you will be able to change with visualization is in this little screwdriver and wrench image, the configuration menu here under Tools. You can pull down Tools and hit Configuration, or you can hit the button directly here in this menu bar. It will pop up a set of things called CC3D preferences, and you will be using, for example, cells, colors. That'll give you a palette to change the colors of your cells, and also 3D. That'll give you some configuration options. Again, when you change these things, you're going to have to hit play afterwards. Cluster borders we're not using at the moment. That's for compartmental cells. Lots of people are pasting good images in the chat. So why don't you try changing the colors of your cells? That would be the preferences menu here. Select cell colors and then select your cell type and then change the color. See if you can do that. Another thing that you can do is you can add additional images to your representation. This button here with two windows on top of each other will add a window. You can also pull down under Windows, you can pull down that menu and select New Graphics Window. And in this case, you can pull the Cell Field menu down and select VEGF. And you will see the chemical concentration. Why don't people try doing that? Okay, the next thing that I wanted to show you is something that we're going to have to do quite a bit. And again, we'll have plenty of opportunity to practice it, but I do need to go through this a little bit before the next session because we're going to need this. We're going to be running a Colonic Crypt Simulation, and that Colonic Crypt Simulation is one that was written actually by a student many years ago. It represents the way the differentiation of stem cells and the proliferation of stem cells in the gut. And the biology is rather interesting. We're not going to have time to go into the biology today. We start with stem cells. They proliferate. After a certain amount of time, there is a chemical gradient in the gut, a molecule called Wnt, that maintains cells at the bottom of the crypt in a dedifferentiated state. The cells signal to each other through contact through this delta notch mechanism I mentioned a few minutes ago. If Wnt is high and internal delta is low, you become a stem cell. If external Wnt is high and internal delta is high, you differentiate into what's called a Paneth cell. If internal Wnt is low and internal delta is low, you differentiate into an enterocyte. And if external Wnt is low and internal delta is high, you become a mucus-secreting goblet cell. And if you get all the way to the top, you die. You fall off and you die from the crypt. That's the basic model. The goal here is not really to do the biology, it's really to understand the model. To understand how to use the model. If you go to the student materials folders you will see a demos for day 1. And I would like you to download to your computer from the Google Drive the Colonic Crypt demo. Now, this is going to be something we're going to have to get used to with CompuCell, which is that model specifications are not a single monolithic file. They are a folder which contains multiple files. And because of that, when we're going to copy files, we're going to zip them and send the zip files back and forth to maintain the file structure. If you are on a Mac, Macs have the unfortunate behavior that if you're not careful, they will unzip folders for you. They will try to guess what you want to do. And that means that you're going to have to either move the containing folder or rezip the files after they've been downloaded to maintain the directory structure. Sometimes you would rather that the computer not be so smart and try to guess what you want to do. And so everybody try downloading the Colonic Crypt zip demo. You'll have plenty of chances to make sure that it works. So if you are running a different simulation in Player, you'll hit Stop, that square box button. And then you will go to the File menu and hit Open Simulation and point to the zipped folder. And it will ask you, will unzip.cc3d project, and hit OK. And you should then be able to run the simulation. Try that. Somebody says, should I download it into the CompuCell3D folder? Put it somewhere where you have write privileges. Your desktop is a good place to put it. You could create a directory called My CompuCell3D in your home directory, but make sure you have write privileges. If you put that folder somewhere where you do not have write privileges, it will crash. Yeah, so I'm going to talk about the 3D rendering next. Now, you can also download with Twedit. You can switch from Player to Twedit by either hitting the little pencil and paper icon here, or under File, pull down menu, Start Twedit, and that will launch Twedit. In Twedit, you can unpack the file, the zipped file, using this up arrow button here, or CC3D project, open CC3D project. In either case, you would select the zipped file, and it'll ask you, do you want to unzip it? And you hit OK. If the file has already been unzipped and you're trying to put it in the same place, it may throw a warning message saying, do you sure you want to unzip it a second time, or do you want to overwrite what I already did? Once you've unzipped it in Twedit, you can select Open CC3D Project, the folder, and then the .cc3d and hit Open. We'll do that in more detail in a few minutes. So this is just to get you familiar with the idea. If you want to see the full file and the code, you double-click in Twedit on the name, and that'll open it up. And if you then want to run it in Player, you can right-click on that and select Open in Player. You can also hit the little CompuCell icon, and that'll run it. All right. So you should see something like this. It takes a while to run this simulation. It's actually rather a complicated simulation. Initially, you'll see blue cells like this. After a while, as people were seeing, they start differentiating. And you're going to see colored cells. And there should be a key which says what kind of cells you have: medium, stem cell, basement membrane, CT, Paneth, goblet, and pterocyte, and so on. Now, if we pull down that XY and make it 3D, you may see a couple of things. You might see a whole green box. You might see something that looks like this. And the reason for this is that in 3D, you have to decide what you want to visualize, because if you visualize everything, you're going to see a box and the cells are opaque. And so you're going to have to decide what cells you want to visualize and what cells you don't. There are a couple of things that you can do. I'm going to jump to here. In this box on the bottom left, you can tell it what cells types to make transparent and what to visualize. In particular, if you're seeing everything as a green box, that's because the BM, which is the material surrounding the tissue that you care about, is being rendered. So uncheck this BM box, and you should see something that looks more like this. What you may see is something that looks like this, where some of the cells are missing. And this is perhaps a bit of an unfortunate thing in CompuCell. And again, you'll have plenty of time to practice this, but I'm telling you about it now, just in case. Which is that CompuCell tries to speed up the rendering by not rendering things that it thinks that you can't see. And in this case, it's guessed wrong. And so there is an option, probably it should be the default, but it's flipped the other way, which allows you to control whether it tries to be smart when it does rendering or not. And to do that, you go to the settings menu here, this screwdriver and wrench. It'll pull up CC3D preferences, select the 3D tab, and there'll be this little button, which is a little bit mysterious, draw outer shell only. Uncheck that box, hit OK, and tell me if you now see the full 3D rendering. Does that work for people? Somebody asked, you can have as many windows as you want. So you can have one window which shows the 3D image and one window which shows the section at the same time. This is only an issue if the rendering is wrong. If the rendering is right, you don't have to worry about it. Yes, it's under Visualization, 3D, Draw Outer Shell Only. Again, if it's working with you, if you can see all the cells, don't worry about this. This is a bug. This is a bug that's specific, maybe specific to the Windows rendering. I wouldn't be too worried about this. This is simply a little bit of a bug check. If you're seeing the 3D rendered properly, you don't have to worry. Okay, Stefan Cini says he sees everything. So if you're seeing everything, you need to go to this menu here and uncheck BM and see if that fixes it. These checkboxes here, where the number 6 is, control what is visualized and what is not. Because you have to, in 3D, you have to be able to look through things. So you need to make certain cells transparent so you can see other cells. So the draw outer shell is a bit of a confusing term. Again, CompuCell rendering the simulation takes a lot of time. And you don't want to render things that are hidden from the viewer. And so CompuCell tries to figure out what will be visible in a particular scene and only render the things that are visible. And sometimes it gets that wrong. And that outer shell is trying to accelerate the rendering by guessing what the visualization is. And if that fails, then you'll only see some of the cells, not all of them. Okay. There's also this cell glyphs option, which instead of representing the actual shapes of the cell, draws little circles where the cells are. That can actually be pretty useful sometimes. Don't get discouraged if this is a little bit of an adventure. We're going to be doing this again and again, and you'll have plenty of time to debug things. One comment, which nobody's talked about so far, but they can happen, which is that the player settings, unfortunately, are hidden. They're in a hidden file called settings.sqlite. And if the player gets into an illegal configuration, you can have a situation where you launch a program and it dies immediately, crashes. Don't delete CompuCell and reinstall CompuCell. CompuCell is never the problem. The sqlite settings, _settings.sqlite, can be illegal. It's not very often, but it can be. And if player crashes when you try to run a simulation, and especially if it's a simulation that ran before or a demo where we know it should work, you need to find the settings.sqlite file and delete it. And that should fix the problem. If you haven't had the problem, don't worry about it. But I want to give you a heads up that that can happen. That would be C users username CompuCell3D _pi .sqlite. The other thing that we've seen today, and it happens especially when you're using Zoom, is that if you open Twedit or Player on an external monitor, and I have a 2-monitor configuration here to run the Zoom setting, and you move Player or Twedit from one screen to another, it will remember where it was. And if you then remove that external monitor and launch CompuCell again, you will see nothing because it will be displaying CompuCell in the wrong place. And that's unfortunately an internal fix in the problem setting issue in the operating system. And so really the best thing to do is that before you finish running CompuCell, if you're using an external monitor and you think you're going to not have the same configuration again, pull Player and Twedit back to your main monitor before you close them. And that's the piece of advice for you on that. Okay. So that's going to be as much as we're going to do today. Again, we're going to have much more time to practice these things. I realize that it takes some effort to go through them the first time. Again, don't be discouraged if you have a few issues at the moment. We will go over things again in the next session and the next. And so you'll have plenty of time to get eased into things. There's a lot to learn. And the goal here is to have you practice things repeatedly so that we can consolidate things. If you had issues with CompuCell, for example, menus not showing up, please document that. Ideally, a screenshot is very helpful.